Next, we have Dr. Kieron Dunleavy, who will be presenting on clinical trials. Dr. Dunleavy is from Georgetown University and the director of the lymphoma program there. Dr. Dunleavy is also a member of our prestigious scientific advisory board. Thank you so much for being here. And after this, we will be having our Q&A session.
Yeah, thanks to Sarah and thanks to the Lymphoma Research Foundation and Dr. Pro and Dr. Muniz for inviting me here today to this great meeting. So over the next 25 minutes or so, I'm just going to talk about what's new in clinical trials in lymphoma and CLL. There really is a huge amount going on, so it's not possible to talk about everything, but I'm just going to talk about some of the major breakthroughs that have happened and some of the things that are happening and hopefully will improve in the future. So what is a clinical trial? So most of the clinical trials I think that we're interested in lymphoma and CLL are looking at the effectiveness of an intervention to treat a disease. And there are many components to a clinical trial. They look at the safety of a new drug. That's usually the first goal. And then the trial looks at how the drug is dosed, and that's complicated, and a lot of testing has to be undertaken in order to identify what the best dose of the drug is that is safe, and then how the drug is effective and what types of diseases it's effective in and what type of lymphoma it's effective in. So we have a lot of drugs, which I'll talk about, which are effective in a lot of different types of lymphomas. So in clinical trials, it has to be worked out where exactly is their effectiveness and which types of lymphomas should they be used in. And then, of course, trials involve exploring combinations of treatments. We have all of these new drugs and agents, and they work very well by themselves, but oftentimes they work a lot better when they're combined with another agent, and there's synergy, so all of these things have to be worked out in clinical trials. There are all types of clinical trials. There are prevention studies in oncology, screening studies, diagnostic studies, quality of life studies, compassionate use studies. But a lot of us are most focused on treatment studies that hopefully aim to improve response rates and improve outcomes for people with CLL and lymphoma. And it's really important, I think, to consider that when we develop new drugs, they really come from work by the scientists that takes a long, long time, sometimes decades. So these drugs, they don't just grow on trees, basically. They come from a lot of thought, a lot of ideas, a lot of hypotheses, and a lot of testing. And very few of these hypotheses or tests actually make it to a drug being developed. But it's in terms of research and work that the Lymphoma Research Foundation supports, the scientific part of drug development is really, really important, because we would never get to the drugs if we didn't have the scientists doing the work in the lab to understand how the drugs work and which types of diseases they're most effective in. And typically, when we test drugs, they're tested in animal studies, and then they go to in vitro studies after that. And there are different phases to clinical trials. Phase 1 is the early part of clinical trial development, when you have a brand new drug and it's being tested for the first time in people. And the focus of phase 1 studies, the first focus is safety, to make sure that the drug is safe to give to people. But often in phase 1 studies, we can see effectiveness as well. So that can lead on to other studies, or it does lead on to other studies if the drug is safe and effective. The function of phase 2 studies is primarily to look at effectiveness. How well does the drug work in the population that it's being studied in? And then phase 3 studies are confirmatory studies, so to confirm the results that were found in phase 2 studies. And then phase 4 is the end part of the phases, when the drug is under FDA review, et cetera. So studies can be conducted by a lot of different organizations, they can be conducted by pharmaceutical companies, they can be conducted by single or groups of investigators. And in the US, the NCI has cooperative groups for adults and children with diseases. So trials can be conducted under the NCI within these cooperative groups. And it's really important to have lots of different groups investigating drugs. So it's important to have industry, it's important to have the government, because they all contribute, I think, very meaningfully to the end product and to hopefully identifying a drug that's going to be effective. There's very careful oversight in drug development to ensure ethical and safe conduct. And each institution that a trial is being conducted in has got an institutional review board, and they oversee the conduct of the trial, and everything is done very carefully. So as Dr. Moon has said, one of the key things in drug development in lymphoma is finding the right target, because if you don't know what you're targeting, then your chance of being very successful is pretty low. And the good news is that we're in an era now where we understand these lymphomas and CLL much, much better than we have done before. I think there's really been an explosion in our understanding of different pathways that turn these lymphomas on or off. So we can go after these pathways with specific targets, and that's really important. This is a bit complicated, but it just shows you the most common type of lymphoma is diffuse large B-cell lymphoma. And 30 years ago, we were not able to distinguish different types of diffuse large B-cell lymphoma. So if someone came into the clinic and they had diffuse large B-cell lymphoma, we could just tell them, look, you've got diffuse large B-cell lymphoma. But through work by many scientists, we now understand that there are many different types of diffuse large B-cell lymphoma. About 20 years ago, we discovered that most of them were called ABC or GCB. But over the last few years, we have even done a better job of identifying more subtypes. And what's important about that is that it leads the way for drug development. So we understand that there are several different subtypes. They're all wired differently. They have different pathways that are turned on and turned off. And that allows us to think about what makes most sense to treat these lymphomas. And then we do clinical trials to answer those questions. So again, this just, I think, tells us how important it is to understand the target and to do scientific work in order to make drug development more likely to be effective. And this also shows you a lot of pathways in lymphoma that are important for targeting drugs. And this is a busy slide, but you can see that a lot of these pathways can specifically be targeted by drugs. So we've talked a bit about the story of the BTK inhibitors, and I think this story is really interesting in terms of how drugs develop. The BTK inhibitors are hugely important in lymphomas and CLL. We now have a number of them that are approved. We have the covalent ones, and now we have the non-covalent ones. But if you go back and look at the history of how BTK inhibitors came about, I think it's really interesting. In the 1950s, there were patients who had a disease called X-linked agammaglobulinemia, and these people had a poor immune system, and they were not able to fight off certain infections. And then it wasn't until years later that the reason that these people are not able to fight off infections was discovered, and it was shown that they had a deficiency in an enzyme called BTK. And then this led to companies investigating this pathway. And actually, they were not thinking that the pathway was important for cancer. They were focusing on non-cancerous conditions. And then Pharmacyclics bought the development pathway, essentially, and then they developed ibrutinib, and after that, many other BTK inhibitors have been developed. So I think this just illustrates that lots of different components go into making a drug, There's a lot of chance. There's a lot of companies and all of the different things that go on in terms of making a company successful. So all of these things are critical for the drug development process in order to develop a drug that's effective for a particular type of lymphoma. This is just one trial that was recently published, and it looked at the outcome for people who received ibrutinib versus placebo who had CLL, and they had bad prognostic features of their CLL, but you can see here that those people who got ibrutinib had an improved outcome compared to those who got placebo. So there have been a lot of advances. The most common type of lymphoma is diffuse large B-cell lymphoma, and a lot of us have been involved in trials over the years where we've been trying to improve the outcomes for patients with diffuse large B-cell lymphoma. And this is an example of one trial that was recently published and looked at a drug called polatuzumab, which we've talked about, which is an antibody-drug conjugate against CD79B, and that was added to the standard treatment R-CHOP. So half the patients got R-CHOP plus polatuzumab, half got R-CHOP alone, and the study showed that there was what's called a progression-free survival benefit of 6.5% in those who got polatuzumab with R-CHOP. And I would say that this is not even a completed story. There are now a lot of other studies that are ongoing to look at this difference and to explore this more and to identify exactly which types of tumors are more susceptible to polatuzumab. And we're certainly not there yet, and there are a lot of other similar types of drugs and non-similar types of drugs that are in the process of being developed based on these results. So it's really an ongoing process, but it's incredibly important for us that we have these landmark studies that show differences in outcome. But also important to know that we're not completely there yet because there's a lot that we still don't understand. And likewise, with Hodgkin lymphoma, this was a breakthrough study where brentuximab, which is also an antibody-drug conjugate that targets CD30, was investigated in people with newly diagnosed Hodgkin lymphoma. And it was compared to the standard ABVD, and there was an improved cure rate for those people who got brentuximab. And actually, at the moment, there is a study that was completed, which is run by the US cooperative groups, which looked at a drug called nivolumab, which is an immune checkpoint inhibitor, and showed an improvement in outcome in people who got nivolumab compared to brentuximab. That's not been published yet, but it may change the way that we treat people with Hodgkin lymphoma in the future. So very, very interesting. Dr. Munez has talked a lot about CAR T-cell therapy. CAR T-cells are very effective in diffuse large B-cell lymphoma. They involve, taking someone's cells and modifying them and then reinfusing them and having them target the tumor very specifically. But the FDA have approved these products both for relapsed refractory diffuse large B-cell lymphoma, but also for other lymphomas as well. And these are some of the trials that have investigated CAR T cells. So, shown here are a number of different strategies that are in development for B-cell lymphomas, and you can see that there are a lot of really interesting targets and a lot of trials that are ongoing, looking at targeting these different parts of the B-cell. We've talked about CD19, CAR T cells target CD19, but there are also other ways of targeting CD19, such as there's an antibody called tafasitamab, which is very effective, and also you can use an antibody-drug conjugate against CD19. The bispecific antibodies are approved for both diffuse large B-cell lymphoma and follicular lymphoma. And these target the B cell, but also target the T cell as well. They target CD3 on the T cells and CD20 on the B cells. These are effective and exciting, and we're at the moment doing studies using these agents in people who've never received treatment, who have follicular lymphoma, for example. So we don't know where the end point indication for these is going to be in certain lymphomas, but they're highly effective, and they might completely change the way that we treat diseases like follicular lymphoma in the future. We just don't know. And then building upon the effectiveness of the bispecific antibodies, there are a lot of other bispecific strategies and trispecific strategies that are in development. So I think the future is very bright. We will probably have even much more effective bispecific or trispecific platforms for indolent lymphomas and, I would say, aggressive lymphomas in the future. And these are being rapidly developed. And then there are a whole host of other ways that the B cells can be targeted. You can target specific pathways. There's something called the tumor microenvironment, which are the non-tumor cells, and we know, especially in follicular lymphoma and in Hodgkin lymphoma, that if you target the tumor microenvironment, you can make a big difference in the tumor, and you can have very good responses when you use that strategy. So that's something that continues to be investigated. And shown here are some other pathways as well. So just for the last part of the talk, I'm just going to talk about MRD in lymphoma. Actually, the Lymphoma Research Foundation just had a big symposium which was focused on MRD and how we're going to move forward with that and how we should be thinking about it in all types of lymphoma. So that took place a few weeks ago, and it was very successful. So this illustrates a dilemma that we often have in when we're treating people with lymphoma. We use PET scans very frequently to stage people when they're newly diagnosed with lymphoma, to assess response, and in follow-up as well, to see if the disease has stayed away or check if it's come back. And this is a patient who had aggressive B-cell lymphoma, and at the end of treatment had this PET scan on the left, and you can see the black area is abnormal. And we look at something called the SUV, or the standardized uptake value, and this was 6, which is higher than it should be. And this patient was observed, and 6 weeks later the abnormality had resolved. But this, I think, illustrates that it's very common for us to use PET scanning in various situations in lymphoma, and while it's very helpful, it can be falsely positive. So we really need other ways of looking at how people respond to strategies and drugs, and also in follow-up, we need to have other strategies apart from PET scanning to check if people continue to be in remission or if their disease comes back. So there's been a lot of work and interest in looking at liquid biopsies, essentially, in all types of cancers, but in lymphoma and CLL, it's been very important over recent years. And there now are a lot of companies who have developed ways of doing liquid biopsies, and there are different things that you can look at in the blood. The earliest types of liquid biopsies looked at something called the VDJ sequence, which is in the immunoglobulin, which is on the surface of tumor cells. And how it works is that the lymphoma is analyzed for this VDJ sequence, which is almost like a unique serial number. And then you can subsequently check for this serial number in the blood. And if it's not there, then that's great. And if it is, it tells you that there is persistent disease there. And that technology has really evolved to the point where not only can you detect if if there's lymphoma or CLL in the blood, but you can also look at genetic abnormalities of the lymphoma in the blood. And that's going to be, I think, very important in the future because we will hopefully have ways of doing a blood test and not only being able to tell if there's persistent disease, but also being able to predict which types of treatments might work just based on a blood test. This, I think, just highlights what this technology or how it can be helpful. And these were people who had aggressive lymphoma, and in the middle of their treatment, they had a liquid biopsy, essentially, and people who were negative by the liquid biopsy had a better outcome compared to those who were positive. And so we know that this technology is very important and very helpful, but I think our struggle at the moment is identifying exactly how to use it. So pretty much all of the big clinical trials in lymphoma and CLL incorporate liquid biopsy, but we're just not at the point yet in the majority of cases where we can use it in the clinic. We can use it in the clinic, but we don't really know what to do with the results. So it's not a good idea to have a test if it's not actionable. So we're really trying hard with clinical trials to figure out exactly when we should be using this test. So hopefully in the next few years we'll have the better standards about when to use liquid biopsy and how it can be actionable, and we're just not there yet. And in terms of the future, in certain types of lymphoma and CLL, we would hope that we're able to identify patients who are cured by early changes in their liquid biopsy or circulating tumor DNA, and also be able to distinguish people who have refractory disease and those who have early relapse, and importantly, before imaging can detect it, and we don't know the answer to that question yet, but could it be that if you can detect a recurrence sooner than it's detected on a scan, might the outcome be better? We don't know the answer to this yet, but we are asking these types of questions and trying to investigate them in clinical trials at the moment. In summary, clinical trials are hugely important in lymphoma and CLL, and the Lymphoma Research Foundation are obviously very much involved in this, and it's really important that they continue to do that and support young scientists and investigators who are involved in the whole clinical trial process. It's really important to consider the scientific part of clinical trials. We never get to be able to develop a drug unless the science is there and really paves the way for it. And that can take a long, long time, and it's really important that we remember to support that, because we'll never get anywhere unless we do that and unless we identify the target. There are lots of components to clinical trials. It's really good news, I think, in lymphoma and CLL that we have such effective strategies, and a lot of our trials now are actually focused on seeing if we can reduce toxicity for people, because the response rates are incredibly high, the remission rates are high, and we're now asking, can we use agents that are not associated with as many side effects as the traditional agents, and that's a really good place to be in as we develop more effective treatments for lymphoma and CLL. So with that, I thank you for your attention. I look forward to our discussion over the next half an hour. Thank you.